Believe I've started recording it. It is okay. Should be recording. So I will go ahead and share my screen and start the presentation. Just to confirm, is everybody able to see my slide as slideshow version?
Yes.
Yes.
So the major points of contact for universal sequencing are going to be myself for technical questions. David Nordell is our inside sales manager, so sort of your day-to-day ordering questions would go to him. Dr. Aaron Levy is our general manager and chief commercial officer, so sort of your higher level business questions would go through him. And Ru Zhang is our director of global distribution. And there'll be some contact info later on in the presentation. Just to, since you're already familiar with us, we'll just go over this quickly. Universal sequencing is a low input, super long read DNA sequencing library prep solution, effectively turning a short read sequencer into a long read sequencer. And there are a number of applications one can use our technology for, and I'm going to spend a lot of the presentation going through various examples and data sets from our assay. Just really quickly at a high level, as you probably are aware, the global NGS market continues to grow. This is from one research study that we had purchased. The demand for NGS continues to grow. And really a big unmet need for the NGS user base is in the long read space. Most of the NGS market right now is short read and mostly Illumina sequencers. And the anticipated need for long read sequencing is around a third of the market. So significant growth in the long read space is expected. And our solution to help bridge this gap is Universal Sequencing's TELSeq library prep. So I'll go through the workflow, but we are a simple, relatively fast one-tube library prep kit that makes your short read sequencer behave like a highly accurate long read sequencer, and also pretty much unique to our solution is that we are a very low input library prep where you're looking at low nanogram or even sub nanogram quantities of DNA for the library prep. And there are a number of applications that I will speak to in detail. Just want to go over at a technical level how we achieve this. And first start with what is likely going to be a review, or at least 2/3 of this diagram should be a review. So we see a representation of diploid genomes being analyzed by next generation sequencing in on this slide. So up top is a representation of short read next-gen sequencing, your standard Illumina libraries, where we are looking at highly accurate reads of 100 to 500 base pairs in length. And this generates many library fragments at low cost and high accuracy. However, it becomes challenging to link any two SNPs of interest to be able to phase haplotypes across an entire chromosome. You really are only able to phase haplotypes if they're within 500 base pairs of one another, which really limits the utility. Similarly, it's very challenging to get any sort of de novo genome assembly with any sort of reasonable amount of coverage with short read sequencing. So Moving down to the middle of the diagram, of course, is the representation of long-read sequencing. And for purposes of this presentation, we're talking about PacBio and Oxford Nanopore longer reads. So these are solutions where a highly accurate read of moderate length of maybe 10 to 15 kb can be generated, or a longer read of lower accuracy, maybe as long as 50 kb. And long read sequencing does allow phasing of some haplotypes, since it's able to capture at least 10 kb of information. So greatly expands both the haplotypes visualized and the ability to de novo assemble a genome. However, there remain challenges in terms of accuracy and also in terms of cost per base pair of information acquired. So our solution to bridging the gap between these technologies is linked reads. So what we do is at the prior to fragmentation of the DNA, we are going to be barcoding the molecules, and we are able to trace a short read library fragment back to molecule of origin based on how we are barcoding these intact very long molecules. And unlike the other two scenarios, we are able to capture molecules greater than 100 kilobases. Most of our data sets will have those very long molecules in them as long as they were present in the isolated DNA. I do also like to point out that in this diagram, you can see there are dashes connecting the library fragments of that originated from the same molecule. We are not a single molecule technology insofar as we are not going to capture the entirety of a given molecule. So I'll go through this on the next slide. We typically capture 20 to 30% of any given molecule, any given single molecule that goes into library prep. So we do still require uh, coverage similar to what you would need for a unlinked short read NGS project. The benefit of our platform is going to be the quality of data and the continuity of data coming out of it. We are not going to be dropping your coverage needs just by the nature of our assay. So I want to spend some time explaining how our assay works. So this is a visual representation of the biochemistry of our library prep. So the diagram moves from left to right. Before I go through the details, I like to point out that our entire assay and library prep takes place in PCR tubes. So there isn't a need for exotic equipment. If a laboratory is already preparing NGS libraries, they likely have all of the equipment and ancillary reagents that would be required to run our assay. So the first step of our assay involves mixing a transpososome complex with our TEL beads and very long DNA molecules. Ideally, customers should be using DNA fragments 40 kb or longer. You can use our assay with molecules shorter than 40 kb. Really, the best data is going to come from starting with molecules 40 kb or above. I will have a figure later on that gives some assembly information relative to DNA molecule size. Again, and with our assay, Around 10 kb is where you really stop seeing a benefit of the linked read technology. So longer DNA going in is going to give a better assembly going out. Do you also want to point out that our tell beads that are going to be critical to the process each have a unique 18 base pair barcode. So that gives us approximately 2 billion uh, barcode possibilities in our pool, so we can handle very complex samples. When we mix all three of these together in the tube, we get the transpososome binding the 18 base pair barcodes, and then semi-randomly binding the very long DNA molecules. What we try to do with our default library conditions is we're going to bind approximately 6 very long DNA molecules to a given bead. So as many as 6 to 8 molecules might share that 18 base pair barcode, and we assign parent molecule identity based on position of the library fragments to its nearest neighbor. So if we have two library fragments with the same 18 base pair barcode, if they map to within 50 kilobases of one another, we assume they come from the same molecule. If they map further or to different chromosomes, then we know they come from different DNA molecules that have bound the same uh, bead. To that, we are going to add a second transpososome complex for tagmentation. So there will be a second transpososome binding, again, in a semi-random fashion to the very long DNA fragments. We then break and wash the DNA, and many of the DNA fragments will have both transpososome complexes bound in the proper orientation, such that we will then take these broken washed beads and PCR amplify, and the molecules that have both transpososomes in the proper orientation will be amplified into Illumina compatible libraries that end up with your standard P7 and P5 adapters. All right. Um, so, a fairly straightforward process. And the reason why we don't capture 100% of the bound molecule is we are amplifying off of where we have the proper orientation of the transpososomes. So in terms of hands-on time and total turnaround time, the assay is fairly rapid. So the actual steps from isolated DNA to sequenceable library fragment is approximately 3 hours. So it is possible for a customer to isolate DNA and go through our library prep and possibly even start sequencing their library all within a single workday. So we are a very uh, easy to carry out library prep. And then I want to talk a little bit about how one would go about analyzing our data. So you take your sequenceable library and put it on a Illumina sequencer and acquire data. What we have in this diagram are sort of two most common paths for data analysis. And the green colorations all are UST developed and maintained software. In blue is 10x Genomics software, which I'll speak to in a moment. So from getting raw data off of the sequencer, the first thing, regardless of downstream analysis wants for the project, customer will run our tell read, QC, and denoising pipeline. This will give cleaned up FASTQ files as well as a QC report with some metrics about the quality of data generated. This output then can be put in to one of two tools by universal sequencing. We have our TEL-link de novo assembly pipeline. This has been optimized uh, for small and medium genomes. We continue to work to make it a more practical solution for larger genomes. So really your genomes up to about 200 megabase in size should work very well with TEL-link. And then for phasing and structural variation analysis of a genome, we have our phasing pipeline, which is called TEL-sort. And currently, both TEL-read and TEL-link are available on BaseSpace by Illumina, and we are working to get TEL-sort released. It will probably be available on BaseSpace in March or April of this year. And our software is also freely downloadable as either Docker or Singularity images from the universal sequencing website, and software could then be run locally on a Linux system. Many of our customers like our software. A good portion of our customers are former 10x Genomics users who do really like that software or are looking to finish out projects where they have already collected and carried out analysis of some samples using 10x Genomics software. So we provide a conversion tool that can be run after running TEL-read. And the conversion tool essentially converts our 18 base pair barcoded libraries into a format that's compatible with the 16 base pair barcode on a 10x Genomics library. And then customers can take those data into either Long Ranger or Supernova, the 10x Genomics legacy software tools. And not really depicted on this diagram is a third option. After somebody runs TEL-read, if they think they have a more clever idea for analyzing barcoded libraries than any of the options laid out in this diagram, there is, again, FASTQ output that could be done with whatever else somebody would like to try and do with it. So just to summarize the workflow, again, it's a 3-hour single tube library prep, so quite easy to deploy. We are a super low input assay, so anywhere from sub-nanogram to as much as 5 nanograms of input for a human-sized genome is required. So we're quite good at scenarios where DNA input might be limited. We've had customers perform de novo genome assemblies using a single insect to get really a truly individualized genome cleanly assembled with very limited material. Additionally, our assay preserves quite long DNA molecule data in its data sets. I'll have some examples where we have molecules greater than 100 kb in length in significant number for some of our data sets that really allows for robust de novo assemblies, scaffolding, and structural variation. I'll have examples of all of that. And our software is, of course, free. Just wanted to, again, call out the low input and kind of compare it to long read sequencers. So again, we're that very low nanogram input, whereas PacBio and Oxford Nanopore tend to need hundreds of nanograms to a microgram of starting material for most of their workflows. So now going to get into what you can actually do with our technology with some actual examples of de novo assembly, preview of our metagenomics tool that's forthcoming, some phasing, SV detection, and then in addition to genome scale phasing, we can also phase targeted panels, which is really neat, and that's what I'll close out with. So like every other molecular biology tool, we're going to start with bacteria and then work our way up from there. So the first bits of data I'm going to show you are from a application note that was authored by Illumina and with significant contribution from universal sequencing. So describing microbial de novo genome assembly, as well using the TELSeq assay. And this was also an announcement of our tools being available on BaseSpace for the backend analysis. So in this application note, Illumina wanted to look at a number of different bacteria with different GC content and different genome sizes. Like to point out on this gel, if you remember from earlier in my talk, I mentioned that 40 kb really is our ideal DNA fragment length. In this case, they were using DNAs closer in length to 20 kb, but they still get really good data coming out. Certainly good enough to assemble a microbial genome, as you'll see. Also do like to point out that we can handle high GC organisms in our assay. The one slight caveat is if it's a high GC organism, you'll likely see a little bit lower library yield since we are amplifying with a PCR step. So there is, of course, the consequence of less efficient PCR if it's a high GC situation. Okay, so there's a lot of numbers on this chart, and I just want to speak to it at a bit of a high level. The first third of this chart is descriptions of the reference genomes used in this study. You can see that genome sizes from about 1.5 to about 5.5 megabase in size, so kind of your standard range for bacteria. And GC content ranging from low GC to high GC, and a number of different plasmid and chromosome number scenarios were looked at here. So we're not just looking at assembling a single chromosome in isolation. In the middle of the table is information about the raw reads coming off of the sequencer. You'll notice that Illumina was collecting 100x coverage here. I think this is their standard coverage depth for unlinked microbial genome assemblies. We find that for most de novo assembly projects, starting at 50 to 60x coverage is often going to give sufficient coverage to get a good genome assembly. This was probably much more coverage than they needed to get the resultant assemblies out that were observed. In that same middle section, you can see reference to SLF size. That is our abbreviation for super long DNA fragment. So this is the calculation from our software of what the initial DNA fragments bound to the beads were. So you can see the DNA fragments were around 20 kilobases going into this library prep. And the bottom really is, I think, the most interesting part, because this is the assembly data. And you can see that for almost every organism, well, for every organism, we were able to assemble the chromosome as a single contig. And generally, we were able to assemble the plasmids as single contigs as well. It got a little more complicated with the most complex sample that was also the highest GC, but we were essentially one chromosome, one contig, and typically assembling 99% of the reference genome for most of the samples. So really a complete microbial assembly using linked reads. And I think I hit the bullet points. Again, good with high GC, really able to assemble discrete contigs. So really getting that long read caliber data out of a short read sequencer. And just to give a more direct comparison of a representative E. coli K12 genome prepared using TelSeq, using a version of Oxford Nanopore, or using unlinked Illumina reads, we can see that the genomic DNA input for TelSeq, again, was half a nanogram, whereas a microgram was required for the other assays. We were able to generate the largest alignment, essentially aligning the entire genome. The highest NA50, again, we were able to link it all together. Since we are using Illumina for the data acquisition, we had fewer misassemblies than the Oxford Nanopore dataset. We also had fewer mismatches per 100 kb than the Oxford Nanopore dataset, and no artificially generated indels, again, because we're using the Illumina instrumentation for data acquisition. So even side by side with Oxford Nanopore, which is often popular in the microbial space, really we still give great data when we are talking about single microbial genomes. We also are very close to releasing a metagenomics focused tool. So here is a preview of that. In this case, we purchased a DNA sample from ATCC that had 20 different bacterial species in it. And this mix was staggered, so there was different relative abundance of these organisms. What we did was we performed our standard library prep, ran it through our TelRead software, and then ran it through our TelLink software to assemble these data into distinct contigs. And then our forthcoming software tool will look at those contigs and assign a strain identity to those genomes. So we're in the process of, we're in the final stages of developing out that software tool. And in this scenario, we were able to not only assemble, but assign identity to the 3 most abundant bins of bacteria. We were able to identify the relative abundance of the remaining species. Just the abundance was too limiting to assign strain identity with high confidence. So this software tool will probably come out early second quarter, I think. Software's coming soon. Want to now pivot to larger genomes and talk about some examples of human genomes. So for these data sets, We're looking at NA12878 and NA24385, two of the genome in a bottle samples. And these were sequenced on NovaSeq's in collaboration with Illumina. So we have on the left, you can see that our library is mapped well to the reference genomes. The majority of the DNA fragments were greater than 20 kilobase in length. And also a non-trivial portion of the DNA molecules in the libraries were greater than 100 kb in length. And that becomes important when we look at not only de novo assembly, but evaluating structural variation in large and complex genomes, as you'll see. Additionally, for the 12878 sample, we had a really good recall rate and precision rate for both SNPs and indels. And we found that a lot of our initial users have been legacy 10x genomics users. So our assay is functionally similar to the no longer available 10x genomics assay. And here's just an example of genome phasing with TelSeq and 10x genomics dataset. And you can see with similar depth of coverage, we were able to phase blocks approximately 3 times the length of the 10x genomics dataset with comparable, I would say, switch error rates. Um, since it's no longer available, don't want to spend too much time on the comparison, but again, you're getting that comparable data quality without having to purchase capital equipment, and it's a faster assay with more barcode diversity than the legacy 10x product. So if you had if you have a customer that used the 10x genomics linked read assay, liked it, they will very likely enjoy using our assay as well. I want to give just a couple of examples of phasing data. So since we're talking about phasing of the human genome, popular relevant region to look at, of course, is the MHC region. And what you can see here is taken from our genome research article. And we are phasing HLA genes, some of the class 1 and class 2 HLA genes. What you can see in this diagram is where the haplotypes were assigned by our analysis. And the coloration is the predicted representative of the predicted a chromosome for a given haplotype. So you can see there are a few mismatches or apparent mismatches where you see orange dots on the blue line. And when we went back and looked at these data, most of these are occurring in lower quality regions of the reference genome. So these may, in fact, some of these are likely a reflection of the actual challenge of this reference genome, and some are just going to be technically difficult regions to assemble or to phase. And then here is another example from our genome research paper of structural variant detection, where we are looking at overlapping deletions in either chromosome. So where one chromosome has a 19 kb deletion and the other chromosome has a 114 kb deletion. So we are able to detect both of these deletions. You could certainly imagine if you weren't able to phase the genome in a robust fashion, you may only be able to pick up the 19 kb region that is missing from both chromosomes. So we are able to phase diploid genomes in medically relevant challenging regions such as the MHC region, and really phase deletions of different size, which would be challenging if you didn't have linked reads. And I want to spend a minute or two on this graph, which is a look at the relationship between phase block size and input molecule length into our assay. So when we say that 40 kb really is where you want to try and shoot for with your DNA isolations, you can see that as the DNA length approaches 40 kb, there's really a significant increase in phased block contiguity, and we observe a similar trend with de novo assemblies. So depending on the sample type, it may or may not be possible to get 40 kb DNA fragments in high abundance. This is also where our recommendation of really you want DNA greater than 10 kb to see much of a benefit. You're not going to be able to assemble significant phase blocks or contigs with DNA below 10 kb. And uh, how long is long enough will, of course, be somewhat customer project dependent. But we do like to show where the desire for 40 kb DNA fragments comes from. So again, another aspect where having long molecules is important is in de novo assembly and scaffolding of your larger genomes. So here is a comparison of de novo assemblies between TelSeq, 10x, Oxford Nanopore. You can see that the TelSeq assay compares favorably with the largest contig, has had the fewest misassemblies, had the largest alignment, and had the largest NA50. And this was a pretty good direct comparison. So it's really, in this case, we were assembling the TelSeq human data set with 10x genomic supernova. So the TelSeq and the 10x data were analyzed in the same fashion. So it really is just a comparison of the biochemistry of our two assays between the TelSeq and 10x genomics data. So I think I hit that yes, we can de novo assemble human genomes with really high quality and great length. But in the non-model organism space, there are really a lot of people using PacBio and Oxford Nanopore to assemble non-model organisms, really build that first reference genome or maybe improve that first reference genome. So not only can TelSeq stand on its own for de novo assemblies, but we can take a pretty good PacBio or Oxford Nanopore genome assembly and further scaffold with TelSeq. So TelSeq will not only have different breakpoints in the assembly because it's a different technology, but also we are taking those 100 kb fragments, really getting some extra long DNA molecules in our library pool to help scaffold other long read data. So even if somebody is using a long read sequencer, they may have and will likely see better data, more complete, more accurate genome assembly if they add on TelSeq data to it as well. And they may find that the TelSeq data is sufficient and sufficiently robust and sufficiently cost effective that it becomes the method of choice for de novo assembly. Yeah, I already went through that. Good assemblies. And and just to mention again, the group that had this PacBio insect genome was the same group that was assembling a whole bunch of insect genomes. So whenever they had sufficient quantities of DNA, they really enjoyed getting assemblies generating assemblies with PacBio and TelSeq together. In the case of where they were sequencing single individual insects and had some very, very small insects and had very limiting DNA, they were able to perform de novo assemblies using a single insect with the very limited DNA, and TelSeq still gave them a genome assembly on its own for these novel insects with single bug. And I want to close out the data set by going over some phased targeted sequencing examples. So you can use TelSeq to phase an exome, which is pretty neat. We have a assay uh, we have an assay out and guidance for built around Agilent SureSelect. So we have a tested and optimized protocol for the SureSelect workflow. You start by generating your TelSeq library in a fashion very similar to how you would generate it for a whole genome application. We just add in some additional PCR cycles to make sure there's enough library for the hybrid capture. Then you go into the Agilent SureSelect workflow. The only modification to the Agilent workflow is we need to use blocking oligos that are made and can be purchased from Universal Sequencing because we have different library priming sites on our adapters versus the standard adapters that are present in SureSelect. So with this workflow, we were able to phase the majority of the exome and also phase the majority of the SNPs present in the sample. And we've found, and it's likely due to the blocker sequence, that we do see the best performance with Agilent SureSelect hybridization captures. You can use our assay with other captures such as IDT or Twist. There may be a slightly lower on target rate with those assays that potentially could be optimized further by the end user. But we can provide the most guidance around SureSelect that, again, we have a protocol and performance expectations around SureSelect. Yeah, just said that. And just one other enrichment technology that we've tested, lab tested with TelSeq is HLS Catch by Sage Bioscience. So this is a initial CRISPR-Cas9 targeting of your DNA upstream of library prep. So that bit of the workflow, so the HLS catch bit would come first, then followed by TelSeq library prep. And in this example, we were able to phase 200 kb regions for BRCA1 and BRCA2. So yeah, another way to phase targeted assays. So those are really the two scenarios that we've tested the most for targeted sequencing. So your hybrid capture and your HLS catch. So for the rest of the slides, I wanted to close out just talking about what it is a customer should buy and what we've seen people most interested in buying so far. So I would say 95% of the time we recommend a new user purchase what we refer to as our standard kit. So there are 4 parts that would be ordered. There is a box of enzyme mixes, a box of beads, and those should be used, and those will be used at the same rate as one another. So our standard volume kit contains enough enzyme and beads for 12 small genomes or 4 large genomes or an intermediate number of intermediately sized genomes. our workflow has 4 different genome target sizes for library prep guidance. The genome size will will determine how much reagent they need. Additionally, we have multiplex, so sample primers for generating your amplicons. And we recommend a new user buy just a single set of multiplex primers 1 through 8. And lastly, the customer needs to use spike-in primers for their Illumina sequencer. So we are compatible with every Illumina sequencer except for the iSeq, which does not accept Illumina spike-in or does not allow for primer spike-in. And we've confirmed that our spike-in primers are not going to interfere with the default Illumina primers. So it is possible to run TELSeq libraries on the same flow cell as and even the same lane if you wanted to as standard Illumina libraries with the standard sequencing primer being used for those libraries. There's not going to be a cross reaction. And here's some list price info for those. Beyond the small volume kit, we do have higher throughput kits as well. These do represent a lower cost per sample, of course, since it's a higher throughput kit. These kits are large enough for 72 small genomes or 24 larger genomes. So it's 6 times the reagent volume. And for the higher throughput customers, we do sell up to 24 multiplexing primers. So if a customer wants to run more than 8 samples on a flow cell, they can. They'll just need additional multiplex primers for sample tracking. And that does point out a current limitation of our workflow is you can only load 24 samples onto a flow cell. This has not proved limiting to date. The only scenario we could really think of, where this might become fewer samples than a customer would want would be if it's a very small targeted assay or bacterial genomes being run on a NovaSeq with an S4 flow cell. But we haven't yet encountered a scenario where somebody needed more than 24 multiplex primers. And just want to pass on the presentation to you. There's more pricing info. David or Aaron, did either of you on yet? No, don't hear that I'm on. Okay. So we have a lead sharing program where I believe David does some work looking at leads in your geography. And if we haven't already provided those, we will do so. Starting this week, myself and our CSO are going to be having a online live Q&A, really targeting prospective customers wanting to understand more about how our assay works. There may be some additional content for those presentations, but right now it's going to be just an open Q&A. And of course, press release and collateral. And for you guys, you can reach out to David at sales@Universal Sequencing for orders. I think we are working on a online-based ordering for distributors as well. The current online order is really more for direct purchase in the US. Wanted to briefly touch on what we've seen have been the customers most interested to buy. So these non-model organism reference genome projects really, as you probably know, like to use lots of technology, different assays to get the most complete version visualization of a genome. So non-model organism groups are great, reference genome consortia, and what have you. Our robustness for microbial identification and characterization also is great, especially those single microbial genomes works really well. And we've seen those 10x genomics legacy users, people that were using the old 10x product before it got discontinued and are looking to finish projects or start new ones in a similar vein, have also been really interested in our products. So these are the three main types of customers we have had to date. And we are seeing increased interest really the world over. And thanks in part to growing our distributor network, we expect the rest of the world to start filling in more on this view, but we do have customers all over. Some additional just technical information. I know I went over quite a bit. Do want to point out that we have various links to more information. Some customer information tends to be of high interest, and those are the links in the middle. The Earth Biogenome webinar contained some of those insect genomes that I was talking about, as well as a Genome Web webinar that was hosted by Illumina and exclusively featured TELSeq data looking at de novo assembly of exotic organisms. Those are two different presentations by the same group, so slightly different depth and spin on their stories. Also, we have our genome research paper, which is one of the most complete pictures of what TELSeq can do to date. And there is that Illumina application note. There's a lot more data than I shared, and it is available online from Illumina, or we can share that with you. Do like to acknowledge that most of the data presented was generated in collaboration with one or more groups, and we certainly appreciate the data that our collaborators helped us generate. And just to close out, again, we're simple one tube NGS library prep that makes a short read sequencer behave like a highly accurate long read sequencer using very little DNA. There are a number of applications you can do today, and as I was giving you the preview of metagenomics, we're always looking to add additional assays and continue to make this good product better. I know I went through a lot and I went through it kind of quickly. I do have time for some questions before we finish up.
Okay, Andrew, thank you. Thank you very much for your presentation. I guess most of the guys already know a lot of from the presentation because we are using it in the lab. So maybe our questions will be more like in a different way, experimental way, let's say.
Okay.
I would say I would have actually two questions. The first question is you've been talking about the compatibility of the TELSeq library prep and sequencing on Illumina with the long read technologies. So the long read technologies creates a backbone for the de novo assembly, and the TELSeq add like extra precision and extra information.
Mm-hmm.
Are you going to, for example, combine it and prepare libraries for example, Oxford Nanopore? Because we definitely do have customers who are using Oxford Nanopore in lab, and maybe this might be interesting for them to actually have library from lower input.
So Right now, I think we're focusing on really Illumina and MGI sequencers. So kind of the next iteration of our assay is, I believe, going to be around modifying our bead barcode to make it perform better with the MGI sequencers. I don't know if we're planning to do any sort of library prep that would directly go on PacBio or Oxford Nanopore. If you think you have a really large opportunity for that and somebody would want to collaborate, that would probably be a question to discuss with Aaron,
Mhm.
or Tom, our CSO, or Ming, our CEO. That one would be kind of above my pay grade. I don't think there are not short-term plans for that. The short-term expansion would be for MGI sequencers.
Yeah, yeah. That was actually the question, like the short-term plans, because so not even, for example, the ion torrent sequencers.
Um, we've so I'm not sure what the data have looked like. We have some people trying our assay with ion torrent. That would be a question probably for our CSO. I haven't been directly involved in
Yeah.
with any of those users to date.
Okay, okay. That's just a specific question because I know about customer who's using ion torrent, and so it's a possibility. I will ask later on. Another thing, we are now thinking of a project with mitochondrial genomes, actually.
hmm
But you mentioned that any fragments or shorter than 40 kb is actually a problem. Could you explain more on that? Why is it a problem if we would like to do targeted sequencing of mitochondrial genomes?
So for targeting, it shouldn't be a problem. The context is really so. I guess with the exception of the last two data slides I showed, everything was in a whole genome context. So for de novo assembly and phasing at the whole genome scale, that's really where the 40 kb is needed. I don't have data in this presentation, but we have done work with shorter amplicons. So it certainly is possible uh, uh, to assemble, stuff in the 10 to even 5 kb length, if it is a targeted application. And that would be something we can uh, give guidance with. Uh, yeah, we've done it.
Okay.
And it just isn't, it's not an officially rolled out assay, but we know how to do it.
Okay, okay. And so connected question is, you do the amplicons. We are thinking of actually hybrid capture,
Oh, okay.
but we are also like thinking of maybe do the hybrid capture actually on the isolates to take the mitochondrial DNA out of the mixture and then doing the TEL-seq library prep. Or do you think it's only possible the vice versa? First TEL-seq library and then targeted enrichment?
It should really be either. So the where was it? This method is sort of is not hype capture based, but it was basically doing that, right? Um, my expectation, just from at least how, and again, this mechanistically different than what you're saying, but kind of conceptually similar. Um, this ends up having a lower on target rate than library prep followed by the capture if you're reversing it, but it would still enrich it significantly. And if you're dealing with such a small sequence, you probably would get, it's okay if you have a low on target rate, um, less critical because you're not having to spend as much sequencing. Um, so it should work. It's just something we don't have specific performance expectations for.
Hmm.
But there's no reason what you're describing shouldn't work. I just couldn't tell you what on target rate to expect, right? And what the yields would be.
Okay, thank you. Yeah, that was my specific question. So I will probably let other place a question if they have any.
I have some. First of all, thank you very much for your presentation. My question or maybe request would be whether it's possible to organize similar session like this and go through the results of Teleread and Telsort and Tellink applications, because I would say that now I can understand the technology,
Mhm.
but it would be good to link it to the output results which we get from our sequencing data. We are doing human sequencing, and right now we are dealing with human genome assembly, and we are somehow evaluating and thinking about what can be done better next day, in the next next run and so on. So
Yeah,
would it be possible to make some direct?
yeah, I think that's something we can do. I will let Aaron know that you guys would like that. So I'm much more wet lab side of things, but our software team has certainly done that before. So I think our kind of like our two main informaticians would be able to certainly have that discussion with you. We can work on scheduling that.
Yeah, perfect. I don't actually think that it's really only for bioinformaticians. Myself also, I'm application scientist. I'm not able to develop a pipeline in command line, but
Okay, so
anyway,
even just like
I can
understanding the
understand that.
outputs
If
of
you
the
want,
QC files and
yeah, exactly. So it would be good to make a connection between what is seen from the wet lab side, what is seen in the report from the bioinformatics, and somehow mix it together and get the full picture, ideally.
yeah, certainly we can I'll talk to Aaron about getting follow up. That would probably be myself, our CSO, and one or more of our software team.
Perfect. Thank you very much.
Maybe I would have some specific question as well.
Mhm.
And it's related to the start of the protocol,
Uh-huh.
which you related to the wet lab part.
Uh-huh.
Because when we are working with the high molecular weight DNA,
Mhm.
it's quite tricky to dissolve the DNA to the requested concentration.
Mhm.
And as you mentioned today, the range is only 3 to 5 nanograms of human DNA, for example.
Yep.
So, I mean, how critical is the highest concentration of the DNA? So, do you have some experience with the, I don't know, let's say 20 nanograms or something like that? Because we have a serious problem to dilute the DNA into the particle such a short range of concentration.
Okay. So yeah, couple of thoughts there. When it's practical, what we've found is to really give the DNA, when you isolate the DNA, let it resuspend overnight, and potentially do serial dilutions, and just give it a lot of time to homogenize. So maybe dilute over the course of a day, over several dilutions, would be one thing. The challenge is it is possible to overload the beads. So really, when we say we want, the 5 nanogram for a human sample, assumes DNA that's between 30 and 30 and 100 kb in length, and that will get you those 6 to 8 molecules per bead. What we'd see if you did 20 nanograms, you'd probably get fewer labels per molecule. So you'd kind of synthetically shorten your data set. Just if we overload the beads, you get DNA molecules competing for space on the bead index transpose on site. So we are underloading the beads, and the reason why we're not saturating the beads is we want to give those very long DNA molecules as much chance to unwind and bind as many sites as possible. Since a lot of the DNA is probably going to be supercoiled, it spatially would be overloading the reaction. So one of the things we're looking at is also, we don't yet have the final guidance yet, but we are probably going to be increasing the reaction sizes for the bead binding steps. We've found promising results in terms of the resultant data. So the actual bead loading conditions are probably going to change sometime in like the second quarter or second half of the year as we're improving the assay.
Okay. Okay.
The total input will probably remain the same. It's just going to be the reaction volume larger. And that would be something that's also when we follow up to go through the QC data. We can go over how you could tell if the beads are overloaded. There's going to be a metric on the QC files. If you're mapping, if you have a reference genome, it'll give you the estimate of DNA molecules per barcode.
Yeah. I understand.
And we expect, if you're getting over probably 10 molecules per bead, you might start to see the data, the assembly lengths shortening, just if we're not maximizing the number of tags per molecule.
Yeah, I'm just asking about the limitations, as you mentioned that how critical is to reach the maximal point of concentration. Because, for example, we have some experience with the tagmentation protocols, and the tagmentation there is working like a normalization of the sample as well. So it would be very, very. Actually, it make all the things much more easier if there would be such a point at the beginning. But thank you for the answer.
Yeah, and like I said, we are, this is really our version 1.0 protocol that, I know we are in the process of making adjustments to make it even better.
Yeah, understand. Thank you.
So do we have any other question, guys?
I don't have any.
No, I don't.
No, thank you.
Okay.
I have a lot of questions, but they are, scheduled for the next meeting.
Yeah. All right, so I'm going to turn off recording then.